3, 2, 1.
The acidification of aquatic ecosystems is an increasing issue resulting from worldwide industrialization and pollution. Burning fossil fuels results in increased carbon dioxide levels in the atmosphere, which can be absorbed by bodies of water.
In water, carbon dioxide dissolves and forms carbonic acid, which is a weak acid responsible for the increased acidity. Acidity is known to be a problem for coral and organisms that contain shells. Not only is acid able to dissolve calcium carbonate, but acidic conditions also lower the amount of calcium carbonate available to aquatic organisms.
With a decreased reserve of calcium in acidic conditions, mollusks are experiencing a decline in their ability to develop their calcium carbonate shells, meaning that the shells are more susceptible to decay and slow growth. Our experimental question addresses the following. How does acidity affect clamshells?
To test this, we set up 3 control tanks with a neutral pH and 3 experimental tanks with an acidic pH. 10 clams were placed in each tank, and each week we measured the weight, length, and width of all clams to record growth over time. Our experiment was run for 5 weeks, and at the end of the 5th week, we tested the strength of the clamshells by recording the weight at which the clamshells cracked.
We hypothesized that clams exposed to acidic conditions have a different shell strength than clams that are in a neutral condition. 60 clams were found in the North River behind the football field at Bridgewater College. The Corbicula flaminia, commonly known as the invasive Asian clam, were our test subjects in this experiment.
At the end of 5 weeks, we tested shell strength by measuring the amount of weight needed to crack the shell of each individual clam. The experimental group included 26 of our 60 clams, 13 from the control tanks and 13 from the experimental tanks. We placed weights on the clam and continued to increase the weight until the shell bore a visible crack.
Figure 1 shows a comparison of the average weight of the clams at the start and end of the experiment.
Figure 2 is the average weight that was stacked on top of the clams until crushed.
According to data collected, there was not a significant difference regarding average weight, length, and width of the clams in the control group and those in the acidic group over the course of the experiment.
All 60 clams had little to no change in shell size. However, Figure 1 does show some variation in the growth rate of the clams. The change in average rate of the clams in the control group is slightly higher than those clams in the experimental group. This difference is not large enough to be significant. Figure 2 shows that there is a difference in the average crushing weight between the control and experimental group. The experimental group had a lower average crush weight of 26.5 kilograms, while the control group had a higher average of 38.6 kilograms. A t-test was performed and it showed a significant difference between the two groups. The calculation resulted in a p-value of 0.0095, meaning that there is a significant difference between the two groups. The clams in acidic conditions did show increased deterioration, since on average these clams had weaker shells that crushed at a lower weight than the clams from the control group. Even though the clams are similar in size for both groups, there is a chance that there were some factors that were not accounted for, such as the age of clams. The clams were not aged before testing, so there could be slight bias in the results because of this. Overall, there was not a significant difference in the change of length, width, and weight of the clams between the two groups, but there was a significant difference regarding the clam shell strength. Therefore, we are able to accept our hypothesis that the clams exposed to acidic conditions have a different shell strength than clams that are in neutral conditions.
Bamber and his contributors performed a similar experiment testing 3 different mollusk species. Their experiment determined that all 3 mollusk species had similar reactions to an acidic pH. As a result of their experiment, clams in a pH below 6 experienced significant mortalities over the course of 60 days. In Bamber's experiment, the mortality of clams in acidic conditions increased as time elapsed. In contrast, our experiment resulted in few mortalities in the acidic tanks. This may be due to the fact that our experiment was not allowed to run for a long enough time. Bamber and his contributors also saw growth suppression, tissue weight loss, reduced shell size, shell dissolution, and suppressed feeding activity that occurred at a pH below 7. Our results did not show significant tissue weight loss or reduced shell size, but we did observe shell dissolution.
Our research contributes to the ecology field by providing information on how the invasive Asian clam is impacted by acidic conditions. This can be applied to understanding how all mollusks may respond in an acidic and freshwater ecosystem. Clams have a significant role in the ecosystem by filtering out toxins that may be harmful to other species around them. The decline of clams due to acidic conditions can have negative impacts to the ecosystem, therefore showing the importance mollusks have in the environment around them.